Hello everyone and welcome back to our forensic podcast. My name is Ain and in today's session I am joined by my friends Shafika, Minza and Aisha. Welcome. So today we are going to talk about something really important and interesting in forensic science, which is postmortem interval or known as PMI.
Yep, and honestly, this topic sounds scary at first, but it's actually very interesting. So, postmortem interval or PMI refers to the amount of time that has passed since a person died. In forensic science, PMI is very important because it helps investigators estimate the time of death and reconstruct what happened before death occurred. PMI is commonly used in criminal investigations to compare the estimated time of death with witnesses' statements or suspect alibis. Besides that, it is also useful in disaster victim identification and missing person investigations. Traditionally, forensic science estimates PMI by observing the physical and biological changes that happens in the body after the death. Some common examples are such as algo mortis, which is the body cooling. We have rigor mortis, which is the muscle stiffening, and livor mortis, which is the blood cooling due to the gravity. Scientists may also observe decomposition stages and insect activity on the body to estimate the time of death.
But Shafika, can you share with the audience why is it important in an investigation?
Yeah, of course. PMI is important because it helps the investigator to narrow down the timeline of death during the investigation. For example, if a suspect claims that they were somewhere else at this certain time, forensic scientists can compare that particular statement with the estimated PMI to see if it matches. PMI also helps the investigators to understand the sequence of events before that happened. In court, PMI findings can become important forensic evidence to support the investigation. So basically, accurate PMI estimation helps the forensic scientists to build a clearer picture of what happened during a particular case.
But Aisha, what is actually happened to our body after death?
So, what exactly happened to our body after death? The heart stopped beating, so oxygen can no longer reach the cells. And without oxygen, cell's membrane cannot produce ATP, that is known as the energy that needed for normal cell's function. So as the ATP decreases, cell's membrane weaken and break apart. Enzymes are then released and begin digesting the body's own tissue. This process is called autolysis. At the same time, DNA and RNA gradually degrade. Next, bacteria from intestine spread to the body and begin decomposition, known as putrefaction. This causes bloating, gas formation, false smells, and tissue breakdown. These molecular changes are important because they help forensic scientists estimate the time since death.
Traditional methods are not always accurate though. Is there any challenges when estimating the time since death using PMI signs like body temperature or rigor mortis like that?
Yes, traditionally, forensic scientists use methods such as rigor mortis, algo mortis, and livor mortis to estimate PMI. However, these methods are not always accurate because many internal and external factors can affect them. So first, rigor mortis is the stiffening of muscle caused by the loss of ATP after death. Its timing can vary depending on physical activities, illness, or drug use before death. Second is the algo mortis, which refer to the cooling of the body after death. However, the body cooling depends on the factors such as room temperature, clothing, body size, and exposure to wind or water. Lastly is the livor mortis that happens when the blood settles in the lower part of the body, causing discoloration. So if the body is moved, the blood pattern may change and may even be mistaken for bruises. Therefore, traditional methods only provided a rough estimation of PMI. Just now, we discussed some limitation of PMI. Are there any new technologies that we can improve in this?
Yes, actually, there's a very interesting new approach called multi-omics.
We keep hearing about multi-omics lately. Can you explain what it means, Miza?
Well, multi-omics integrates data from various molecular layers such as genomics, transcriptomics, epigenomics, proteomics, metabolomics, lipidomics, and microbiomics to comprehensively map the biochemical and microbial changes that occur after death. By tracking processes such as protein degradation, microbial succession, and metabolic shifts, multi-omics provides more precise and individualized insights than traditional methods as mentioned before. One of the key strengths of multi-omics integration is its ability to address the limitations of single omics approaches, which often capture only a fraction of the complex postmortem changes. So, for example, metabolomics can monitor the accumulation of postmortem byproducts, while proteomics identifies patterns of protein breakdown linked to specific time intervals. Likewise, transcriptomics and genomics uncover alterations in RNA and DNA, revealing the body's molecular response to environmental changes like temperature or trauma. Microbiomics, the study of microbial communities active during decomposition, has emerged as another key area, offering unique insights through the dynamic patterns of microbial succession. So this integrated data helps forensic scientists to construct a detailed molecular profile, which is valuable for cases with severely decomposed or burned remains. So these multi-omics platforms rely on cutting-edge technologies such as mass spectrometry, next-generation sequencing, and advanced bioinformatics tools to process and analyze vast datasets to uncover meaningful patterns and correlations. These computational approaches enable the researchers to identify key biomarkers and construct predictive models for PMI estimation with high precision. So, for example, machine learning algorithms can analyze patterns across omics layers to generate comprehensive timelines of postmortem changes. The integration of these datasets not only enhances the accuracy of PMI estimation, but also sheds light on the molecular dynamics underlying postmortem decomposition. Although multi-omics integration is still a developing area in forensic science, its potential to refine PMI estimation is undeniable. By bridging the gaps between traditional methods and molecular approaches, multi-omics is paving the way for more accurate, reliable, and scientifically grounded forensic investigations.
What about cell-free DNA? Isn't that also being used now?
Yeah, that's true, Shafika. According to the research that I have come across recently, scientists are trying to see whether cell-free DNA can be used to estimate the time since death. According to the study, the scientists use rats as the sample, and they will be grouped into different time since death, from 0 hours to 48 hours. And then, after each time since death, scientists will collect the blood sample from the rats, and they will do extraction to obtain the cell-free DNA. Next, they will do qPCR, or known as quantitative polymerase chain reaction, or in simple words, it's like you are amplifying the DNA to measure the concentration level of DNA in the sample. And then after they got the result, they noticed that the concentration level of cell-free DNA in the sample decreased over time. So this method is very useful compared to the traditional method, because just now we have discussed traditional method can be challenging due to the several factors, right? But cell-free DNA is different, because after death, the cell-free DNA will be break into several pieces, and the changes in the fragment size can be used to estimate the time since death precisely.
But multi-omics can be expensive. How do we apply it to every crime scene?
Besides traditional methods, forensic scientists are now studying modern multi-omics approaches such as genomics, transcriptomics, proteomics, and metabolomics. These techniques provide more detailed information about molecular changes after death. However, they also have limitations, including high cost, advanced equipment requirement, and the need for trained experts to analyze complex data. Another important limitation is time. Traditional PMI methods can provide results within minutes or even hours, but multi-omics analysis often takes much longer because of the sample preparation, laboratory analysis, and data interpretation that are very complex. Therefore, although multi-omics methods are highly promising and scientifically advanced, their practical use in real forensic investigation is still limited by cost, time, and technical challenges.
I see. So more advanced doesn't mean perfect, right?
Honestly, this has been a very interesting discussion, and I feel like we could talk more about it, especially with how fast forensic technology is evolving today. But sadly, we have come to the end of our discussion. So to conclude our discussion today, traditional methods such as algo mortis and rigor mortis are the common approaches in estimating the time since death. However, this method could be challenging due to the environmental conditions such as changes in temperature, humidity, insect activity, and also the conditions of the dead body itself. So now scientists are moving towards modern technologies such as genomics and transcriptomics. And one of the examples is cell-free DNA. Although multi-omics can be expensive and there are so many ethical considerations that needs to take into account, but it remains promising for future research if we study this further. So thank you so much for joining me today, and I will see you in the next video. Bye bye.